Okay, so welcome to the the continuation sa atong chapter 4, which is the topic of microbiology. Okay, so we are now here sa atong common laboratory ng mga procedures. Okay, so here you will be discussing aseptic technique. Aseptic technique, ibig sabihin, is a method where you perform sterilization or preventing contamination sa imong ginaobra. Ay a practice whereas you will reduce your contaminants. Para kung mag-inoculate ka sa imong culture media, ang imong ginapatubo lang gid nga bacteria ang mag-grow. Okay, aside sa septic technique, which is your mga sterilization to sa aton nga atong ginabalta nga gina-autoclave ang aton nga media, ang aton nga agar plates. Okay, the septic technique is procedure sa laboratory gid. Okay, the application na. Okay, so here you will be working with many pathologic species of your bacteria in a laboratory. Remember that bacteria are also in the air, also sa aton nga skin, sa counter, sa aton nga table ng ginobrahan, okay, sa aton nga biosafety cabinet, okay, and also other mga objects kag mga equipment nga ginagamit mo that have not been sterilized. Okay, the most important tool of transferring your culture is your wire inoculating needle or loop, duwa na sila kabilog. Can be quickly sterilized by heating it in a Bunsen burner. Okay, ang imong tip sa aton nga loop o needle dapat maging red hot na siya to ensure nga mapatay ang bacteria nga present. Okay, adjust the air sa aton nga Bunsen burner dira. For those nga wala pa kakita sa Bunsen burner, okay, ano yung tsura. Okay. Okay. This is a Bunsen burner looks like. Okay, tignan sa picture. Pero mas may color naman. So, Bunsen burner here. So, makita nyo nito sa dilang side. This is your valve or regulate sa aton flow sang gas. Okay. Then, so that there are hotter aton nga inner cone and cooler sa outer flame. Ibig sabihin, kung mag-flame ka sa babaw sang aton nga inner cone kagat sa tunga sang outer nga flame. Okay, a dry needle may be sterilized by holding it a 30-degree angle. So this is the 30-degree angle. So yung mga kamot, you will be holding it like that. Okay, in the outer part of the flame. A wet loop, okay, with bacteria on it should be held in the inner part of the flame, okay, to avoid splatter. Ang ginamin niya na. Okay, after mo na inoculate or butang sang imong bacteria halin sa imong broth or sa imong ginkuhaan, which is a liquid, a broth, culture broth. Okay, so simply may basa-basa pa ng gamay. Okay, kung para sterilize mo, para mapatay ang ginsaylo mo na sa imong culture media, okay, ang magbutang ka sang imong loop, okay, sa inner nga flame. Okay, may picture dito. Right. Na, okay, you have your images. Na, you have here the inner flame and this is your outer nga flame. Okay, kung magbutang ka sang loop mo, nga wet na, hindi ka sa tunga. Okay, ginamin niya to avoid splatter, hindi magpisik-pisik naman. As, kaya diba, kung mag-boil ka sang init na tubig, okay, may chance na mag-splash. Therefore, kung mag-splash siya, at least may outer flame ka pa nga mag-burn sa iya. Therefore, to avoid infection. Okay, so ang mga ginahandle. Okay, then heat until red hot gihapon to ensure gid nga wala sang may nabilin. Okay, always flame the loop immediately before and after gamit. Pero when you're using your loop and also your needle, okay, use i-flame mo gina siya. Okay, the flaming itself is the aseptic technique. Okay, then allow it to cool before picking up a inoculum or mga bacteria mo, inoculum of your bacteria naman. Because kung tamaan pa na siya kainit, kung kapula pa siya, then gintusmaw na siya sa imong broth. Okay, aside sa nga way kasi may makuha, may mapatay ka pa nga mga bacteria mo sa imo nga culture nga broth. Okay? Pwede mo kung nga matos mo muna siya para migon mo daw siya sa imo nga broth, but it is not advisable naman. Because usual na nga katapik da is the mga dead na nga mga bacteria. Therefore, hindi magtubo sa imo nga culture medium. Okay? Then, always remember, okay, always stand a carry tube in a rack. Okay nga man. Para at least may tungtungan ka dayon kada tapos mo gamit, hindi mo pwede ginakapta ng imo nga culture broth. Okay? Okay, you will be transferring your hand. Okay. Try ko makakita sa YouTube. Okay. Continue lang. Okay, do not lay the cup down or touch anything with it. You will hold your cup, okay, sa other mo nga hand. Okay, kung face to face kita, you will be performing a practical when it comes sa aton nga septic technique. Next is do not remove the lid or unnecessary for a prolonged period of time. So kung tapos mo nakuha sa imong nga bacteria gamit ang mga loop, make sure mo sarado mo na siya dayon. Pero before that, you close the lid, dapat gina-flame mo na siya. You have here your loop. Always put the lid face down. You have here the connecting loop in para dahatya. Then you have your broth, culture broth. Gina flame mo siya sa babaw. Okay, to ensure nga wala sang may kung may magwa man da nga aerob nga aerobic. Okay, aerobic ba yung term? Ang bacteria nga pwede ka like airborne na check na term. Ang airborne nga bacteria, okay, mapatay siya sa flame. Okay, not necessarily nga ibutang mo ni sa ang tube mo sa kalayo, but close enough sa flame. Okay, then you have here ang imong ginpainit nga loop. Okay, kuha ka na sa imong nga culture broth. Silo mo na sa imong nga plate. Next, i-flame mo siya liwat to ensure nga wala sa may makasulod di aside sa imong culture da. Okay, you have two purposes to prevent airborne nga bacteria makagwa and also prevent man airborne nga bacteria makasulod. You have here, i-hit liwat, then dira ka na mag-close. After mo na inoculate, also yung mga loop nga gingamit, you hit it again para reusable. And you have here how to streak for isolating colonies. Okay, septic technique. I have here. Okay, hindi siguro dugay Tasig lang. While we wait for it to load, we have here how to streak for isolated colonies. Okay, streaking. Okay, you have here streaking for isolated colonies. Okay, you have here mixed cultures. More than one species can be isolated using a streak plate technique. Okay, the goal is to acquire a pure culture of one species of your bacteria in a single colony. From a mixed culture, we do this to separate the microbes on the surface of the agar with a quadrant streaking. This method dilutes the bacteria, and the goal is not to have bacteria in all of our quadrant but to get a single isolation of a certain nga colonies ng mga bacteria. Okay, you have here the streaking method. Okay, first, kaya di ba nag-streak ka to dito sa mga, you have here, an example. Okay, the only one you streaking is dili nga side lang. Okay, after that, begin close mo na, di ba close mo lang. Then also hit mo siya liwat. With the use of that certain loop mo liwat, okay, kaya mo siya, you get a colony of bacteria here, streak mo, so this is your second quadrant. Okay, then hit mo liwat yung mga loop, hulat ka mag-cool down, then streak ka liwat for the third quadrant by pulling that through area sa halin sa mga second quadrant. Then you have there the fourth quadrant, which is halin naman sa third. Okay, kuha ka gamay. Okay, this is an isolation. Okay, strict for isolation colonies or isolated colonies. Ginapanubo
In this
ang
video, you will
iyang
learn the proper
quantity
practices and
to
procedures
have
for
a more
performing aseptic technique.
separated nga
Using aseptic technique, you will be
culture
able to
growth.
transfer a
Okay,
bacterial monoculture into
kaya
a
nagaload
fresh
na ko.
medium
So this is 6
without introducing any
minutes nga
contaminating
aseptic
microorganisms.
technique video. Okay, so sa
Two of the
Biorad
most commonly
Laboratories.
used
In this
tools
video,
for
you will learn
transferring
the proper
bacteria
practices and
are the
procedures for
inoculation
performing
loop
aseptic
and the
technique.
inoculation needle.
Using aseptic technique, you will be able to transfer a bacterial monoculture
Knowing how
into
to
a
use a
fresh
Bunsen
medium
burner is an
without
important
introducing
skill
any
in aseptic
contaminating
technique.
microorganisms.
To light a Bunsen burner, start by closing the valve at the base by
Two of the
rotating the
most
collar
commonly
so
used
that the
tools
fats
for
inflate.
transferring bacteria are the
Then slowly turn on
inoculation
the
loop
gas.
and the
Once you
inoculation
hear the
needle.
gas, ignite the burner and set the height of the flame by adjusting the amount of
Knowing how to
gas.
use a Bunsen burner is an
The
important
temperature of the
skill in
flame can be
aseptic
adjusted by
technique.
rotating the collar
To light a
control
Bunsen
the
burner,
flow of
start by
oxygen.
closing the valve at the base by rotating the collar so that the vents are
The
covered.
coolest flame is yellow and orange.
Then slowly turn on the
The
gas.
medium flame is blue.
Once you hear the
And the
gas,
hottest
ignite the
flame is a
burner
roaring
and
blue
set the height of the
flame
flame by
characterized
adjusting
by
the
a
amount
clear
of
blue
gas.
cone in the middle.
The
The
temperature
tip
of
of
the
the cone
flame
is
can
the
be
hottest
adjusted
part
by
of the flame.
rotating the collar to control the flow of oxygen.
Inoculation tools are sterilized by passing them
The
through the
coolest
hottest
flame
part of the
is
flame.
yellow and orange.
Once
A
sterilized, the
medium flame
inoculation
is
tool
blue,
should not be set
and the
down on
hottest
any
flame is a
surface.
roaring blue flame characterized by a clear blue
In
cone
this
in the
section,
middle.
you will learn how to transfer
The tip of the
bacteria
cone
from a
is the
liquid
hottest
culture
part of
to
the
a petri
flame.
dish. After flaming a loop, take the lid off the
Inoculation
container of
tools are
liquid culture
sterilized by passing
and
them
pass
through
the
the
mouth through
hottest
the
part of the
flame.
flame.
Then
Once
dip the
sterilized,
loop into the
inoculation
liquid
tools should
culture
not be set down
and
on
flame
any
the mouth
surface.
again before replacing the lid.
In this section, you will learn how to transfer bacteria from a liquid culture to a petri dish. After flaming a loop, take the lid off the container of liquid culture and pass the mouth through the flame. Then dip the loop into the liquid culture and flame the mouth again before replacing the lid.
Okay, so as you can see here, diba, sa pag-handle niya sa cup class, okay, with the use of its right hand, okay, if right-handed ka, usually ang imong needle, you handle it like a ball pen, okay, pagbukas siya with the use of its pinky, okay, then ang
Transfer
cup,
the
ara
bacteria
lang
to a
sa
petri plate.
iyang pinky.
Lift the lid of
Tandaan
the
nyo na
plate and
ang
hold it over
gawa
the
mo na
base as a
proper
shield
procedure
to
kag para
prevent
at least duha
contaminants
po
from
sa
falling
inyo
onto
para
the plate.
mag-cover liwat.
Spread the
Kay
bacteria
makuha
over
ka
the
lang,
entire
tapos na mo
surface
siya
of the plate
kadali,
in all
hasta nga
directions.
makatubig na diing imong loop.
Rotating the plate a quarter
Okay?
turn between
Then,
spreads will ensure that the
hit mo liwat na
bacteria
yung
is
mga
spread
bottle, then
evenly
close
across the
mo.
plate.
Transfer the bacteria to your petri plate. Lift the lid of the plate and hold it over the base as a shield to prevent contaminants from falling onto the plate. Spread the bacteria over the entire surface of the plate in all directions. Rotating the plate a quarter turn between spreads will ensure that the bacteria is spread evenly across the plate.
Okay, so as you can see here, sa akong na-explain kina, wala sa nagamit sang isolation nga technique. Okay, but spreading niya lang gid sang isa lang gid ka brought yung cultured na. Okay, so this is just only video description or video demonstration sa atong nga aseptic technique but not the streaking nga technique. So, everything clear ha? Okay, kaya mas mambal nga. Lain man niya ginaubra, lain man niya gina-perform. Aside that, kung maka face-to-face na kita guro by December man, hoping, we will do directly the practical. Okay, practical kids. So, individually or by group, okay, you will be performing this. Hoping that we will see each other sa atong mga face-to-face.
In this section, you will learn how to transfer bacteria from a liquid culture to a liquid. After sterilizing an inoculation loop, take the lid off the container of liquid culture and pass the mouth through the flame. Then dip the loop into the liquid culture and flame the mouth again before replacing the lid.
In this
Now take the
section, you will
tube
learn
of
how to
liquid
transfer
media and pass
bacteria
the
from a
mouth through
liquid
the flame.
culture to a liquid. After sterilizing an inoculation loop, take the lid off the container of liquid culture
Then dip
and
the
pass
loop
the
with
mouth through the
bacteria
flame.
into the tube.
Then dip the loop into the liquid culture
Flame
and
the
flame
mouth
the
of the
mouth
tube
again before
before
replacing
replacing
the
the lid.
lid. Now take the tube
Flame
of
the
liquid
loop to
media
sterilize.
and pass the mouth through the flame. Then dip the loop with bacteria into the tube.
In this
Flame the
section, you
mouth
will
of the
learn how to
tube
transfer
before replacing
bacteria
the
from a
lid.
petri plate to a stab culture. Start by sterilizing an inoculation needle
Flame
and
the
passing
loop
it
to
through a flame.
sterilize.
Allow the needle to cool. You can test for coolness by tapping the needle in a clear area on your auger plate. While holding the lid as a shield,
In this
gently
section, you
rub
will
the
learn how to
inoculation
transfer
needle over a
bacteria from
single
a petri
colony.
plate to a stab culture. Start
Transfer
by
the
sterilizing
bacteria
an
to a culture
inoculation
tube
needle by
filled with
passing it through
nutrient
a
agar.
flame.
Remove the top of the tube and insert the needle,
Allow the
making
needle to
3
cool.
deep stabs.
You can test for coolness by tapping the needle in a clear area on your auger plate. While holding the lid as a shield, gently rub the inoculation needle over a single colony. Transfer the bacteria to a culture tube filled with nutrient agar. Remove the top of the tube and insert the needle, making 3 deep stabs.
So as you can see here, gamit siya sa aton nga deep agar sa aton nga bottle. Okay, gamit naman sa galing
When
sa
finished, replace the
needle.
lid on the tube
Okay,
and
naman
sterilize the
because
needle.
this type of agar promotes an aerobic environment naman. Ang
In
matubo
this
nga
section, you
bacteria
will
is
learn how
not
to
sa
transfer
top
bacteria
sa
from a
aton nga agar
petri
but
plate
with
to a liquid.
inside sa aton nga agar nga tube.
Sterilize an inoculation loop by passing it through a flame.
When finished, replace the lid on the tube and sterilize the needle.
Let the loop cool. You can test for coolness
In
by
this
tapping the
section,
loop
you will
in a
learn how
clear
to transfer
area on your
bacteria
auger
from a
plate.
petri plate to a liquid.
While holding the lid as a shield,
Sterilize an
gently rub the
inoculation
inoculation
loop
loop
by
over a
passing
single
it through a
colony.
flame.
Now take the tube of liquid media, remove the lid, and pass the mouth through the flame.
Let the loop cool. You can test for coolness by tapping the
Then
loop
dip
in a
the
clear
loop with
area on
bacteria
your auger
into the
plate.
tube.
While holding the lid
Pass
as
the
a
mouth of
shield,
the container
gently
through the
rub the
flame before
inoculation loop
replacing the
over a
lid.
single colony. Now take the tube of liquid
Sterilize the
media,
inoculation loop
remove
to
the
complete the
lid,
process.
and pass the mouth through the flame. Then dip the loop with bacteria into the tube. Pass the mouth of the container through the flame before replacing the lid. Sterilize the inoculation loop to complete the process.
Okay, so demonstration sa aton nga septic technique. Okay, so if with further discussion here, mga procedure, you can just use YouTube for a quick demonstration. Okay, and you have here types of your media. Kato ganyan na, you have your dip, diba? You have your slant, your slant dip. Okay, ang difference between your slant, the slant is halin dili gid asta. Okay, sa gid sa corners ang imong mga tube. Then you have your slant butt, your slant dip nga agar, a tube. Then you have your dip and your broth. This is a liquid. Then this is a broth with a door hub tube. Okay? What is the purpose of this door hub tube? Tandaan nyo niya, the door hub tube is uh, to know if ang imong nga bacteria nga gina-culture dira will produce an oxygen or may bubble formation. Okay? Because some certain nga mga bacteria, okay, ang ilang after product or waste product is oxygen or i-excrete nila na, okay? Kaysa nga i-release nila okay, may mga bacteria nga already will be releasing the oxygen here sa Durham tube and it will form bubbles. Okay, so you will need to uh, measure the amount or the length or the size of the bubble form. Then you have here plates. Okay, the plates damdam usang type of culture plate. Okay, there's too many to mention and too many to memorize. Mga basic lang aton kwaon since you will not be handling health or mga pathologic nga mga bacteria pareha sa akong course, but more lang sa introductory. You have your slant, okay? So, kita nyo na din, this is slant medium with agar various nutrients and also mga indicators. Indicators na because either ang agar mismo ma-change color depending on what type of bacteria is growing. Yeah, that's why they call it indicators. Your slanting gives a bacteria a greater surface area in which to grow in a tube. Okay? Aside man daan yung mga plates, yung mga indicator man na, mga indicator type ng mga plate. Okay? Then you have your agar slants are also useful in maintaining bacteria cultures. More so that than a stack of petri dishes. Masapos mag-store gani. Magtipon, mag-preserve, or kung pabili mo na sila as source for laboratory experiments. Okay, or ang imong mga tube. Okay, because you will have a test tube wrap. Yung mga test tube wrap depende kung ano kadamo ang pwede niya ma-handle kaysa sa agar plate that is very consuming, okay, sa space kag sa imong nga agar mismo, kag mas budlay i-handle kag mas secure ang mga tube nga man you will be having a cap, okay, securing sa unod ya than your agar plate which is separated nga duha ka glass covers. Then you have here multiple cultures are placed in the test tube racks and stored under or inside the refrigerator. Bacteria are inoculated into a slant using a loop and grow in the surface of the agar. Then you have your dant or slant dip, okay? A solid medium made with the agar of various nutrients and indicator, do similar lang. But ang shape lang nila ang naglain. Okay, thus it is similar but creates a deep zone commonly called your bud. This type of culture medium gives the ability for the bacteria to grow, okay, to grow in both aerobic and aerobic, which is the surface here, and also anaerobic kung i-pinch mo sa o mag-inoculate ka with the use of a inoculating needle. Okay. Ayan, explain ko na. Including stabbing needle which it plays tweaking across the surface sa atong slant. Next, you have your dip. Okay, so medium agar with various nutrient indicators. Same. This type of culture is used for the growth of anaerobic. O, tama ba ko? Same procedure with the use of a needle. Then your broth, liquid medium. Okay, na-explain ko na. Okay, kung broth, okay, makita mo kung ano nakadamo ang bacteria nga gagrow, okay, based on its turbidity. Turbidity is the cloudliness, okay, gaburon siya. Okay, makita nyo yung mga particulates nga form, that is your bacteria colonies forming in a broth medium or a liquid medium. Okay, and bacteria is inoculated into a broth using a loop. Okay, you will be using a loop. Okay, ang bilog yan na, it will daw dira magsulod ang tubig nga may bacteria mo. Next is your broth with a Durham. Na-explain ko na to. Your Durham tubes are CO2 galing hindi oxygen. Okay, oxygen ginagamit ya, CO2 or nitrate ang iya nga byproduct. Okay, the tube is essentially filled with a medium collects gas that bacteria grow, creating the bubble. Then bacteria is inoculated into broth. Gusto ang tube ang gihapon. You will common sense na lang niya class, hindi ka pwede ka gamit sang needle with kung magkuha ka sang mga liquid naturally. Okay nga amad, wala sa may matapik. Okay, may matapik but small quantity. Therefore, it will not be enough or tugay siya katama mag-grow kung liquid to liquid ka magpatubo sang imong nga microorganism. Okay? Then you have your plate. Isolate medium with made by your agars. Nutrients and indicators can be made in a petri dish and various sizes. Plates are particularly helpful in isolating specific species of your bacteria which is not possible in a liquid medium. Using the Amo to ang aton nga streaking for isolated culture technique. Bacteria can be diluted until individual colonies can be formed. Bacteria are nucleated into a plate using a loop. Okay, then you have here, please be aware that the loop will collect much higher concentration of bacteria from a plate than from a broth. Okay nga aman. Okay, sa aton nga broth, you will have your the water mismo, ang iyang broth, then partially lang na ang mga bacteria. Pero with the use of a plate, okay, ang kwaon mo niya sa plate is the colony na gid yan mismo. Okay, ang tubo na gid sang imo nga bacteria. Therefore, you have a higher concentration of your bacteria compared with your broth. Okay, then while using a CIF technique, please consider that the media or medium you are talking with what you are taking from the bacteria from. Then your aseptic transfer of bacteria, this is now working individually. Okay, nakita na nagina sa video. Okay, ang explanation, din-explain naman to. Then you have here bacterial shapes and arrangement. Discuss ko na to sa aton nga starting sa aton nga starting sa aton previous nga video or video presentation sa aton nga topic. Then you have your preparing of an emulsion. Okay, this is emulsion. Ay, so
Okay,
may ma-search sa
Assalamualaikum and
YouTube.
very good
At least
morning.
diretso
My
na
name is Bob
with the
Iskandar.
use of
I'm a lecturer in my course
ito nga
okay,
video.
in Agrarian
Tapos,
Health Science. So for this time practical, I'm going to share with you with the preparation of emulsion. So according to the people formula preparation, the ratio of 4 water. In this case, the
media
protocol
prep,
on emulsion,
preparing
the
of
doctor
emulsion.
requested us
Okay,
to
so baba. Let's see if this is sa aton nga bacterial related or base sa
Okay,
chemistry.
Assalamualaikum and very good morning. So my name is Bob Iskandar. I'm a lecturer in my course, okay,
It's
in
a video
under Faculty
from
of Health
Course
Science.
Official.
So for this time practical, I'm going to share with you with the preparation of emulsion. So according to the people formula preparation, the ratio of 4 water.
Okay, this is chemistry ni sa
In
gid.
this case, the cod liver
Okay,
oil emulsion, the
chemistry
doctor
ni,
requested
not
us to
for microbiology. Okay nga man. Because sa aton microbiology will be having an emulsion lang for aton nga slides. Okay? At slides ta para ma-prepare mo siya for staining and fixation. Wala nga term. Okay, wala sa aton nga YouTube. So, hoping face to face, makita nyo gid personally kung paano ginaubra. Okay, so working in pairs ni sa dapat. Then, with the slide, gamit ka sa wax and pencil or labeling tape, then pencil. Okay, do not use a Sharpie. This will be the bacterial concentration in one area outside. Okay, prepare an emulsion on each side. If you are taking bacteria from a plate, place a small drop of water. For clarification, you will not be using a tap water class. It's either you'll be using NSS or normal saline solution or ano ba yung tubig? Nature spring. May term din ang water yung ginagamit sa lab which is sterilized na gid, di man filtered. Yung nalipat lang sinsya gid. Denatured. Nadula lang. Lang sa tips na akong baba. And aseptically add your bacteria. If you're taking bacteria from a broth, okay, make sure you place 3 to 6 loopful of bacteria into a slide with no water added. Kung ano na kung broth ang imo, pero kung gagamit ka sang plate which is you will be getting a colony, okay, you need to add water or your NSS. Then if your lab partner will need to prepare the following for the slides. Okay, you have 3 slides for Staphylococcus epidermis. Hoping by sa aton nga may nakita laboratory equipments and also mga bacteria culture na prepared. You have 3 slides of your Pseudomonas aeruginosa and 2 slides for your Staphylococcus epidermidis and Pseudomonas aeruginosa mixed with together for a gram stain as one backup. Okay, then mix gently until it will make a cloudy mixture. Then, hindi ka pwede ka mix gid taman ka aggressive naman because you will lose the bacterial morphology. Anong morphology gani nila? It's either wait, ang class Okay, so let us continue. Okay. Now, allow the slide to air dry. Okay, on the side warmer. While the slides are drying, start your negative state. Okay, once the liquid is completely evaporated, and then you need to heat fix it. Okay, heat fixing is where you will kill what na discuss ni. Heat fixing is where you will kill the bacteria. Okay, para at least stable na siya. Okay, wala na sa hindi siya mag-mix or hindi siya magtubo sa imong nga staining reagent nga kaysa usually sa laboratory dan ginadip na siya sa mga ilang bottles. Okay nga provided. Pero sa iban man it's either uh, gina gina-flow, ginabutangan sa slide mismo. Okay? Then by the use of your fire, okay, initon mo or itaw siya sa babaw sa mga flame 3 times. Okay? If you heat too little, the effect will be the bacteria will wash off sa ating slides. Naman, aside sa ginapatayan na ang mga bacteria, okay, ginapastik yan na ang imong smear or ang mga emulsion sa imong glass slide. Okay? Then if you heat too much naman niya, okay, you will cook the bacteria and denature them. Ibig sabihin, madula gid sila. Completely. Wala na sa amin dilin man sa imo. Okay? Allow the slide to cool and then continue with your staining protocols. Now you have here your negative stain as what I discussed previously. Your negative stain with the use of your Negocin black. Okay, it is a simple and indirect stain that used for the determining of your bacterial morphology lang. Okay, then the shape and the sizes of your organism is seen as a color-free outline against the dark background. Okay, the advantage of this using this method is that prior to the fixation of your by heat, okay, wala na kinahanglan. So the organism are seen in more lifelike shapes. Your negocin is an acidic stain which becomes negatively charged. Then, with the surface, most of your bacteria, which is positive, which is positively charged, bacteria are negatively charged. Okay, kay negative ang imong nga stain. Then, wala siya ga-absorb sang imong nga stain. Kay negative man yung mga bacteria, negative man yung mga dye, so wala sila ga-stick. Ay ga-repel sila. Therefore, the glass of the slide will be stained, giving you a black background or a negative background. Okay, but the bacterial stain cells will not. Okay, if you need to step outside and watch this video, we have here a link sa ating video. And then add a small drop of your negocin to the slide. Okay, aseptically transfer a loop full of your negative stain. Mix bacteria into the drop of negocin and mix gently. Use a second slide held in a 45-degree angle to smear across your slide. Okay, this is what you call your smearing. Okay, smear na is sa mga droplet ipa ang angle. Paano ko ni-demonstrate with the face-to-face man. Let me try research that on the internet. Smearing. No. Oh, pitching. Smudging. Wait. Nice. We have here pictures na. Smearing class is the way of spreading the imong emulsion nga which is one drop lang sa imong slide. Na akong summary explanation kasi review. Wala gid ang akong ginapangita explanation or demonstration. Okay, then allow it to air dry on the counter. Do not use the slide warmer. Then repeat the procedure to make a slide of your Staphylococcus epidermidis and your Pseudomonas aeruginosa. Then examine it. Then butang ta sa aton nga laboratory manual or aton nga worksheet. Next nga example aton nga staining is your simple stain. Okay, simple stain nga man, you will be using your reagent which is your methylene blue. I explain ko na ni. You have here more elaborate kag procedural introduction ta na with the use of the slide that is already nga gin-prepare mo na. Okay, okay, add your methylene blue to heat fix the slide and leave it for 2 minutes. Okay, hold the slide in an angle and rinse with water. Okay, gently lang class ha. Do not hindi taman kakusog gid ang imong water para hindi mag-wash off ang imong smear da. Then hold the slide squirt with the bottle. So, gamit mo yung water. Pwede man sa faucet. Okay? Then, okay, amo na gali explanation pagid aside da. Okay, why you are now pwede ka na kagamit sa mga tap water with the maghugas na sa mga smears mo. Okay? Because ga-wash off ang mga bacteria nga arada. Aside pa sa naga-wash off ang mga bacteria nga arada, ang imo nga smear mismo, okay, is already stained na. Bacteria mo will have a color, yung mga background will have a color. Therefore, ang mga buhi ng mga bacteria sa atong mga tap water will not be that much visible na sa aton nga smear nga prepared. Naman, because you will not be watching the slide nga basa pa siya sang tubig. Okay, ipa-air dry mo pa na siya, pamalhon mo siya, then dira mo siya ibutang sa imo nga microscope. Okay, so while the main point is while doing the laboratory, okay, you need to make sure kung mag-culture ka, hindi contaminated para sigurado gid nga kung anong gina-culture mo nga bacteria nga da amo lang gid na magtubo. Okay, hindi ka pwede gamit sa mga tap water lang. Okay, nga wala na pa boil na filter. Basta ang term yapon nga sa distilled distilled water usually ginagamit sa aside sa chemistry sa ato man nga microbiology sa distilled water. Okay, distilled gaman because napabukalan na siya to the level nga wala na gid sang microorganism nga arada. Okay, then blunt it gently sa tissue paper to get the excess nga water. Then examine under your OIO, which is the highest magnification sa aton nga microscope. Then you have next here your gram staining. Okay, you will be examining the morphology, yung natura. Okay, then you have your gram staining. Okay, the definition of your gram stain. Okay, most important is the differential stain. Samaray na lang because I've been repeating this. Textual kita sa aton nga procedure. Kung paano kita mag-ubras ang gram staining. You have here a picture of your gram stain or your microorganism. Nga gram positive and gram negative. Gram positive, they're color blue, they are purple. And for gram negative, they're either pink, reddish pink, pinkish red. Okay, same na, but they are not orange. Okay? It is either pink, red, or orange, or pinkish red. Na. So you have here, same yung mga procedure sa mga spare nga Staphylococcus epidermidis, Pseudomonas aeruginosa, and your Staphylococcus extra are mixed together sa mga tilgali. Kaya mixture of your Staphylococcus epidermidis kag Pseudomonas aeruginosa. Because they are two different species of your bacteria, therefore they will have a different morphology and also ilang gram staining. Okay, procedure sa ating gram stain. Okay, stain your smear with your crystal violet. Okay, with pabutang mo sa imong crystal violet, you need to time it for a minute. Okay, after that you rinse it with water, then treat it with your iodine mixture. Okay, ang aton sa laboratory na it is grams iodine. Okay, for 1 minute liwat. Ang ginamin niya ng 1 minute, let it soak sa aton nga dye for 1 minute. Babayaan mo na siya. Then, rinse with water liwat. Okay, after you rinse it, you need to decolorize it with alcohol. Okay, decolorizing alcohol. Full term niya. Okay, then wash it with water liwat. Okay, ang ginawa brade, it's either 3 to 6 drops, pero sa among laboratory practice as much nga wala nagawasang bilin nga crystal violet, because the crystal violet will give the color blue nga dye sa aton nga cell. Then, after the decolorizing, kaya wala na siya blue, or kung may mga naging gram positive na da siya, okay, ang mga gram negative na wala ga-take up sa nga mo nga dye will be washed off completely. So, you will need a counter stain. A counter stain, for 30 minutes, you will be using safranin. Pero sa full ganyan, safranin red. Red din man, safranin red nga na to. Just purely safranin. Then, rinse it with water. This is your counter stain. This will give the red color of your gram-negative nga mga bacteria or microorganism. Okay, proper gram stain is mixed cultured and worth 2 extra credits. Mga bonus points lang. Over here, procedures. Okay, crystal violet will give a blue color. Then, your grams iodine. It's color brown. Okay, iodine. Then, you have your decolorizing alcohol. This will wash off the mga wala ka take up sang gram positive. Okay, then your safranin as your counter stain. I-stain niya ang mga gram negative. Okay, kaya ang mga gram positive, atong gani atong explanation na sa iyang peptoglycan. Okay, silang hindi sila magbaton or hindi sila magpaspasulod sang sang imong crystal violet ang imong gram negative. Okay, therefore, ma-wash off siya sa imong decolorizing. Then, ang imong kay may color na imong gram positive, ang imong gram negative naman. Okay, what will happen is mga common mistakes here. Okay, wala ka naging makita. Okay, wala ka ka-fix mayo, nag-wash off siya. Okay, aside that, everything I see is purple. Okay, wala ka nag-decolorize mayo. May mga bilin-bilin paghihapon sa imong crystal violet. Then, everything I see is pink. Okay, nag-decolorize ka tama nga mas kini mo gram positive nga mga bacteria pati ang ilang aday nga na wash off. Next is your Acidfast. Okay, lapit naman natin acid na lang. Where was I? Okay, here. Okay, your Acidfast staining or AFS, Acidfast staining. Okay, this for the use of mga after you will have your spore nga stain and also capsule nga stains. Okay, you have Acidfast. Acidfast stain is a differential stain that used to identify acidfast organisms such as the member of your genus Mycobacterium. Your acidfast organisms are characterized by a wax-like, nearly impermeable cell walls. They contain a mycolic acid and a large amount of fatty acids. Okay, the common sense na lang class that the way you counter yung mga fats na is with an acid. Just like an example, if you are eating too much fatty foods, di ba, namin magpatunaw, therefore you have a soft drink which is more like an acidic effect. Okay? Pero suppose mo matunaw yung mga kinaon. Di ba? Because of their fatty acids, mga waxes, or mga complex lipids. Lipids is another term also for fats. Okay, this type of cell wall is resistant to common compounds. Therefore, acid-fast organism requires a special staining technique. Okay, the primary stain used in your acid-fast stain is a carbol-foxin. Okay, it is a lipid soluble and contains phenol. Okay, which helps the stain penetrate the cell wall. Okay, this is further assisted by the addition of your heat in the form of a heat steam. Kaya nga no is, while may ang mga slide, may carbofloxacin, kaya ang mga smear, okay, ginapaano mo siya sa init. Okay, you are putting it above a burner, pero sa among laboratory, why na kami ka burner na, we are just using a lighter. Okay. Same lang naghihapon. Basta as long kami mainit mo lang siya hasta nga mag-bubble sa kadali. Okay? Okay, that's the bubbles. Okay, it is considered as a steam. Okay, or mag-asosa. Okay, then steam helps to loosen up the waxy layer and promotes the entry of your primary stain inside the cell wall. Then the smear is then rinsed with a very strong decolorizer, okay, which strips the stain from all the non-acid-fast cells but do not permeate the cell wall of your acid-fast na organism. Okay, ibig sabihin ano pa lang ito na color blue la. Okay, ang napa-color blue ya pa lang is your mga non-acid-fast nga mga bacteria. Okay, ibig sabihin mga acid-fast mga bacteria, which is the main purpose of this method or yung this technique, is wala pa stain. Okay, after mo na decolorize, okay, mga non-acid-fast does not permit the cell wall and of the acid-fast organism. Okay, the decolorizer of acid-fast cells then take up the counter stain, and which in the case it's your methylene blue. Sorry again. Okay, ang naglasa lang ako. Okay, correction na lang. Okay, your carbofunctionally it's the one who gives the red color. Then your methylene blue, which is your counter stain. Okay, or sa imo man lang din nga simple stain. Okay, so your the one who gives the non-acid fast nga mga color. Okay? Kaya ang imong gina-hunt di sa aton nga method is your color red nga mga or mga kung baga sa gram staining is your gram negative. Okay? So, is ang acid fast. Okay, this is the opposite of your gram stain. Okay? Kasi if you're talking about acid fast positive, okay, you are looking at a red nga microorganism, and if you're talking about mga non-acid fast, it's either color blue sila. Ano na? You have here mycobacterium or bacterial transparent prior your staining. Then with the use of your carbofuncin, it gives a pinkish or red color. Then after mo decolorize ang imong mycobacterium, which is a acid fast bacteria, okay, ay mga non-acid fast will give a light pink. Then after mo na mabutang sa ang mga counter stain, which is yung methylene blue, okay, ay yung mga non-acid fast lang. Okay, here sa procedure, okay, just skip it na. Okay, so maintindihan kung mag-perform yung kag face-to-face it. Now, para mas madasig atong discussion, so we are now on spore stains. Okay, spore stains, okay, in the spore, hindi na lang, video na lang kung okay, quite complicated siya. Slightly, may mga acid fast. Acid fast staining
is used to differentiate and classify the bacterial organisms in
microbiology.
which is the causative agent of the tuberculosis, the TB in humans and animals,
Here,
and
a video demo, okay, from microbiology and biotechnology.
the causative agent of in solution. They can easily take up the stain of the tuberculosis in animals. Certain bacteria
My job
on
is to
application
make
of
college
the
easier
carbofuncin
because
staining
students
solution, they can easily take up the stain
Let me
to their
have a
cell
skip
flush.
that.
Next, on application of the decolorizing agent, the acid alcohol, they can be easily get decolorized
Bacterial
and
organisms.
leaving the bacteria
Bacterial
colorless.
organism, the gram-positive.
On
Okay.
contrast,
The
certain
acid
bacteria
fast
on
pathologist,
application of the
Friedrich Nielsen.
carbofuncin staining
So,
solution,
may
method
brief
is
they are
discussion.
difficult to
popularly
stain.
well known as Zeal.
But
Nielsen's
once they
technique.
take up the stain, further
This
on
is a
application
differential
of the
bacterial staining
decolorizing
method.
agent, the
Here,
acid alcohol,
more than
they are
one
not
stain is
decolorized,
used.
and
This
leaving the
technique
bacteria
is used to
stain
differentiate and
pink.
classify
Such
the
type of
bacterial
bacteria
organisms
are called as the
Okay,
acid-fast bacteria.
okay, or
The other group of
TB
bacteria
sa mga
humans
dots,
which
and
get
TB
easily
dots,
decolorized
TB
with
dots
acid
ang hindi
alcohol
napangita
are
nila
called
usually.
as the non-acid-fast bacteria.
the causative agent of
The very good
solution.
example of
They can
the
easily take up the
acid-fast
stain.
bacterium is
Cation of
mycobacterium.
the para
These
in
mycobacteria are
animals.
slender rods.
Certain
The
bacteria
surface
on
of this
application of
mycobacterium
the
consists
carbofuncin
of
staining
a liquidal
solution.
wall
They can easily
called
take
as
up the stain
glycolic
to their
acid,
cell
which is a
surface.
waxy material over the
Next, on
mycobacterial
application
surface.
of the
So,
decolorizing
because
agent,
of this
the
liquidal
acid
wall,
alcohol,
these
they can
bacteria,
be easily
on
get
application of
decolorized
carbofuncin
and
staining
leaving
solution,
the
they
bacteria
are
colorless.
difficult to stain.
On
But
contrast, certain
along with
bacteria
the
on
continuous heat
application of
treatment,
the
they take up the
carbofuncin
stain
staining
by
solution,
passing through
they are
this lipoidal
difficult to
wall.
stain.
Further,
But
on
once they
application
take
of
up
the
the
acid
stain,
alcohol,
further on
the
application
decolorizing
of
agent,
the
they are not
decolorizing agent,
decolorized
the acid
and
alcohol,
leaving
they
the
are not
bacteria to
decolorized
stain
and
pink.
leaving the
This is called
bacteria
as
to
the
stain
acid
pink.
fastness.
Such type of bacteria are called as the acid fast bacteria. The other
In
group of
general, this
bacteria, which
acid-fast
get easily
bacteria
decolorized
cannot be
with
stained
acid alcohol,
easily.
are called as
However,
the
once stained, it
non-acid-fast
cannot be
bacteria.
de-stained.
The very good example of
This
an
acid-fast
acid-fast
bacteria
bacterium
are
is
generally
mycobacterium.
arranged in bundles.
These
This is
mycobacteria
due
are
to
slender
the presence
rods.
of the waxy material,
The surface of this
mycolic acid, over the
mycobacterium consists
mycobacterial
of
surface.
a lipoidal wall
Ultimately,
called
this
as
gives a
mycolic
bundle of
acid,
faggot
which is a
arrangement.
waxy material, over the
This is
mycobacterial
the
surface.
bundle of faggot.
So,
The
because of this lipoidal
mycobacterium
wall,
will
these
have
bacteria,
this type
on
of
application
arrangement.
of carbofuchsin staining
So,
solution,
the
they
arrangement
are
of the
difficult to stain.
acid-fast bacteria is called as
But,
bundle of
along
faggot
with the
arrangement.
continuous heat treatment, they take up the stain by passing through this
Coming to
lipoidal
the
wall.
principle behind the
Further,
acid-fast
on
staining
application
technique.
of
This
the acid
technique
alcohol, the
differentiate the
decolorizing
bacteria
agent, they
based
are not
on their
decolorized
coloring
and
property.
leaving the bacteria
Therefore,
to
we will
stain
discuss
pink.
their coloring
This is
property
called as
of
the acid
the
fastness.
acid-fast
Such
and
type of
the
bacteria
non-acid-fast
are called
bacteria
as
in
the acid
parallel
fast
to each
bacteria.
other.
In
Once the
general,
bacteria
this
are
acid
treated
fast
with the
bacteria
strong
cannot be stained
carbofuchsin, which
easily.
is the primary stain
However,
used here,
once stained,
along with
it
the
cannot
heat
be
treatment, it gives a
de-stained.
pink color to both the
This
bacteria,
acid-fast bacteria
but
are,
only
generally,
the
arranged in
acid-fast
bundles.
bacterium
This is
fixes
due
the
to
primary
the
stain.
presence of
Next,
the waxy
when
material,
the
the
acid alcohol
mycolic
is
acid,
added,
over
this
the
is the decolorizer.
mycobacterial surface.
It decolorises
Ultimately,
the
this
primary
gives
stain
a bundle of
easily
faggot
from the
arrangement.
non-acid-fast bacteria.
This is
But
the
the
bundle of
acid-fast
faggot.
bacteria
The
retains the
mycobacterium
primary
will
stain.
have this type
Lastly,
of
on
arrangement.
use of the counter stain,
So,
the
the
Leffler's methyl
arrangement
and
of
blue
the
that
acid
gives
fast
blue
bacteria
color
is
to the
called as
non-acid-fast
bundle of faggot
bacteria into the
arrangement.
background. So, on acid-fast staining, the
Coming to
non-acid-fast
the
bacteria in the
principle
background
behind the
appears
acid-fast
blue,
staining
and
technique.
the
This
acid-fast
technique
bacteria appears
differentiates the
pink.
bacteria
This
based
color
on
difference is
their
due
coloring
to their
property.
difference in their cell
Therefore,
wall
we will
constituents.
discuss their
The
coloring
surface
property
the
of
acid-fast
the acid
bacteria
fast
consists
and
of
the
lipoidal
non-acid
wall
fast bacteria
called
in
the
parallel
mycolic acid,
to each other.
which is a
Once
waxy
the
material
bacteria
over
are
the
treated with
mycobacterial
the strong
surface.
carbofuchsin,
But this
which is the
waxy
primary
material
stain used
is
here,
not present
along
over
with the
the
heat
non-acid
treatment,
fast
it
bacteria.
gives a pink color to
So,
both the
on
bacteria,
microscopic
but
examination,
only
the
the
background
acid fast
appears
bacterium
blue, and
fixes the
the
primary
acid-fast
stain.
bacteria
Next,
stains
when
pink.
the acid alcohol is added,
But
this is the
on use
decolorizer.
of the
It
counter stain, the
decolorizes the
malachite
primary
green
stain
staining solution
easily from
that gives
the non-acid
green
fast
color to the
bacteria,
non-acid-fast
but the
bacteria
acid fast
into the
bacteria
background.
retains the primary
In
stain.
that case,
Lastly,
on
on use
microscopy,
of
the
the counter
background
stain,
appears
the
green,
Leffler's methyl and
and
blue
the
that gives
acid-fast
blue
bacteria
color
stains
to the
pink.
non-acid fast bacteria and to
The
the background.
demonstration.
So, on acid
The
fast
procedure
staining,
for
the
the
non-acid fast
acid-fast staining
bacteria in the
technique.
background
The
appears blue,
materials
and
required
the
are
acid fast
the strong
bacteria
carbofuchsin
appears
staining
pink.
solution,
This
which is
color
used as a
difference is due
primary
to
stain
their
in this
difference in their
experiment.
cell wall
The acid
constituents.
alcohol
The
used as a
surface of
decolorizer.
the acid
This
fast
is prepared
bacteria
by using
consists of
3%
lipoidal
hydrochloric
wall
acid and
called
95%
the mycolic
alcohol,
acid, which
and
is a
the
waxy
Leffler's methyl
material
and blue
over the
staining solution,
mycobacterial
which is
surface.
used as a counter
But
stain.
this
In this
waxy
experiment,
material is
these
not present
are
over
the three
the
important
non-acid fast
solutions
bacteria.
required for conducting the acid-fast
So,
staining
on
technique.
microscopic
Apart from
examination,
this,
the
the other
background
requirements
appears
are
blue,
the
and
forceps,
the acid fast
the
bacteria
blotting paper,
stains pink.
the spirit lamp, the
But
distilled
on
water,
use of the counter
and
stain,
the
the
smear
malachite green
prepared from a
staining
sample,
solution
which
that
is
gives
air-dried and
green color
heat-fixed.
to the
And
non-acid
lastly,
fast
the
bacteria
staining rack.
and to the
These
background.
are the requirements for
In
conducting
that case,
the
on
acid-fast staining
microscopy, the
procedure.
background appears green,
First,
and
prepare
the
the
acid
smear
fast
from
bacteria
sample.
stains pink.
Spread the smear to the
The demonstration. The procedure for the acid fast staining technique. The materials required are the strong carbofuchsin staining solution, which is used as a primary stain in this experiment. The acid alcohol, used as a decolorizer.
size
This
of
is
about
prepared by
1
using
centimeter
3%
square,
hydrochloric
so
acid
as
and
to form
95%
a thin
alcohol.
film.
And the Leffler's methyl and blue staining solution, which is used as a counter stain
After
in this
spreading,
experiment.
allow it
These
to air
are
dry.
the three important solutions required for conducting the acid fast
It may
staining
take
technique.
2 to 3
Apart
minutes
from this,
for drying.
the other requirements
After
are
air drying,
the
fix the
forceps,
smear by
the blotting
heat
paper,
by passing over the
the
flame
spirit lamp,
2 to 3 times
the distilled
with
water,
a slow sweeping
and
motion.
the smear,
This is called
prepared from
heat
the sample,
fixation of
which
smear.
is air-dried and
After
heat-fixed.
the
And
preparation of
lastly,
smear,
the staining
place the
rack.
fixed smear
These are
on the
the
staining rack.
requirements for
Then,
conducting
ignite
the
the
acid
spirit
fast
lamp.
staining procedure.
First, First, add
First,
the strong
prepare the
carbon
smear
fuchs and
from
staining
sample.
solution over the
Spread the
smear.
smear to the
Okay, so ang klase ng bakot sa inyo na smearing kasi sa video. Yan sa smearing pagdi gani.
Cover
Mas
the entire smear with the
kuha
staining
na
solution.
ang Ganun na.
Heat the
Halin
stain
din
until
sa plate.
vapor just begins to
Tapos maiwan
rise.
isa.
Do not overheat.
Size of about
Allow the
1
heated stain
centimeter
to remain
square
on the slide
so
for 8
as to
minutes.
form a thin
Great care
film.
must be taken when heating the carbofixin. Only a small
After
flame should be
spreading,
applied
allow
under the
it
slides
to air
using an
dry.
ignited spirit lamp. After 8
It
minutes,
may take
allow the
2 to
slide to
3
cool
minutes
down,
for drying.
and then wash
After
it
air
with
drying,
the
fix
distilled
the
water.
smear by heat by passing over the flame 2 to 3 times with a slow sweeping motion. This is called heat fixation of smear. After the preparation of smear, place the
Secondly,
fixed
add
smear
the
on the
acid
staining
alcohol,
rack.
the
Then
decolorizer,
ignite
over the
the
smear.
spirit lamp.
Cover the entire smear
First,
with the acid
add
alcohol,
the strong
and
carbofixin staining
allow it
solution
to act
over
for
the smear.
2 minutes or until the
I-soak
smear is
nyo gid siya.
sufficiently
So,
decolorized to
di lang
pale pink.
sa matraver lang as the smear, but
After
the
2
whole
minutes,
slide.
wash it with the
Cover
distilled
the
water.
entire smear with the staining solution. Heat the
Lastly,
stain until
add
vapor
the
just begins
Leffler's
to
methyl and
rise.
blue
Do
staining
not
solution
overheat.
over the smear. Cover the entire smear
Allow
with
the
the
heated stain
staining
to remain on
solution,
the slide for 8
and
minutes.
allow it to stand for
Great care
30
must be
seconds
taken
to
when
1
heating the
minute.
carbofixin. Only
After
a small
30
flame
seconds
should
to
be
1
applied under
minute,
the slides
wash it
using an
with
ignited
the
spirit
distilled
lamp.
water.
After 8 minutes, allow the slide to cool down, and then wash it with the distilled water.
Distilled water ang nakita ko nga term kayo na.
Secondly, add the acid alcohol, the decolorizer, over the smear. Cover the entire smear with the acid alcohol, and allow it to act for 2 minutes or until the smear is sufficiently decolorized to pale pink. After 2 minutes, wash it with the distilled water. Lastly, add the Leffler's methyl and blue staining solution over the smear. Cover the entire smear with the staining solution, and allow it to stand for 30 seconds to 1 minute. After 30 seconds to 1 minute, wash it with the distilled water.
Okay, so additional note. Okay. Nga sa mga methylene blue, kaya nga daw sa tunga lang or sa smear lang. Because sa itong
After washing,
carbol
blot
foxide,
dry
di
the
ba?
smear
Kita niyo man ito
using
ginang
the blotting paper.
heat mo siya. So if you're heating it, may chance nga lessen nga ma-lessen ang mga carbol foxine, mga dapat entire slide mo gid siya. Di ba kung ma-heat mo, ma-evaporate ang liquid, therefore mabuhina na yung mga staining solution. Okay? So kay sa imong methylene blue, sa imong counter stain, wala mo naman siya ginahit. Okay? So ang imo na lang nga smear area, ang imo nga make sure mo nga na soak or nabutangan gid sang imo nga counter stain. Then also make sure niyo nga matandaan niyo gid kung diin nga
Now,
part
the stained
sang
smear
imo
is
nga
ready
slide
for
ka
the
nag-smear.
microscopic
Okay?
examination.
Di ka basig malipat ka
The
basig sa other
microscopic
side sa gali.
examination.
Okay? Just need to
Place
make sure
the stain slide
nga ang imo
over the
nga
stage.
naka-face after me is ang imo nga
Examine
slide
the slide
nga may
under
smear.
100x oil immersion. Objective microscopy. On
After
microscopic
washing,
examination.
blot dry the smear using
Following
the
the
blotting paper.
Acidfast staining technique. These Acidfast bacteria are stain pink, which are slender
Now,
rods,
the
Okay, so dito
and they are
nakita niyo
arranged
king
in
na.
a bundle of faggot
Okay, sa
arrangement.
blotting paper
Due
or
to the
tissue
methyl and blue
paper.
counterstain,
Okay?
the background
Ang
is
gina-wipe nila
stained blue.
nga daw
This
entirely is ang
background gives a good
opposite. But
contrast
if you are
for the Acidfast
trying
bacteria in pink.
to
So,
dry
the
it
result
with the use
is
of
written
your
as
tissue
the smear
paper or
prepared from the
blotting
sample
paper,
stained
itong
using
pag-blot is
the
ito
Acidfast
gina-tap
staining
niya lang.
technique is
Okay?
positive for
I-tap
the
lang
Acidfast
gid. Do not
bacteria
wipe
with
your
the bundle of
smear.
faggot
Klaro
arrangement.
ha? Kung
The
isu-wipe mo na
example
siya,
of bacteria is the
focus ginada
Mycobacterium
tanan.
species, which possesses this type of microscopic feature on Acidfast staining technique.
Now, the stained
Coming
smear
to
is
the
ready
summary of
for
the
the
Acidfast
microscopic
staining
examination.
technique.
The
First, the
microscopic
preparation
examination.
of the smear
Place
from the
the
sample.
stain slide
After
over
smear
the stage.
preparation, air dry, and heat
Examine the
fix
slide
the smear.
under
After
100x
fixation,
oil
add
immersion,
the strong
objective
carbofluoxetine staining
microscopy.
solution
On
over the
microscopic
smear.
examination,
Heat the smear with the staining solution
following the
for 8
Acidfast
minutes.
staining technique,
After 8 minutes,
these
allow the
Acidfast
slime
bacteria
to
are
cool down,
stain pink,
and
which
then
are slender
wash it
rods,
with the
and
distilled
they are
water.
arranged in
Next,
bundle of
add
faggot
the
arrangement.
decolorizer,
Due to the
the
methyl
acid
and blue
alcohol,
counter stain,
over the
the
smear.
background is
Allow
stained
it
blue.
to act for
This
2
background
minutes,
gives a
or
good
until the
contrast
smear is
for the Acidfast
sufficiently
bacteria in
decolorized
pink.
to pale
So,
pink.
the result
After the
is
smear
written
is
as
sufficiently
the smear,
decolorized,
prepared from the
wash it
sample,
with the
stained
distilled
using
water.
the Acidfast
Lastly,
staining
add
technique
the counter
is
stain,
positive
the
for
Leffler's
the
methylene blue
Acidfast
staining
bacteria
solution
with
over the
the
smear.
bundle of faggot
Allow it to
arrangement.
stand for
The
30
example of
seconds to 1
bacteria
minute.
is the
After 30
Mycobacterium
seconds to 1
species,
minute,
which
wash it
possesses
with
this
the
type of
distilled water.
microscopic feature
After
on
washing,
Acidfast staining
blot dry
technique.
the smear using the
Coming
blotting
to
paper.
the summary
Now,
of
the
the
stained
Acidfast
smear
staining
is ready
technique.
for the microscopic
First,
examination.
the preparation
Place
of
the stain
smear
slide
from
over
the
the
sample.
stage.
After
Examine
smear
the slide
preparation, air
under
dry,
100x
and heat
oil
fix
immersion.
the smear.
Objective
After
microscopy.
fixation,
With
add
this,
the
we are
strong
coming to the end
carbofuchsin
of
staining
the
solution
Acidfast
over the
staining
smear.
technique.
Heat the
The
smear
detailed
with the
protocol for
staining
the
solution
Acidfast
for 8
staining
minutes.
technique is
After
available
8
as
minutes, allow the
downloadable link
slide
in the
to
below
cool down,
YouTube description.
and then
In next
wash it
video
with the
demonstration,
distilled
we will
water. Next, add the decolorizer, the acid alcohol, over the smear. Allow it to act for 2 minutes, or until the smear is sufficiently decolorized to pale pink. After the smear is sufficiently decolorized, wash it with the distilled water. Lastly, add the counter stain, the Leffler's methylene blue staining solution, over the smear. Allow it to stand for 30 seconds to 1 minute. After 30 seconds to 1 minute, wash it with the distilled water. After washing, blot dry the smear using the blotting paper. Now, the stained smear is ready for the microscopic examination. Place the stain slide over the stage. Examine the slide under 100x oil immersion, objective microscopy. With this, we are coming to the end of the Acidfast staining technique. The detailed protocol for the Acidfast staining technique is available as downloadable link in the below YouTube description. In next video demonstration, we will...
Okay, so that will be all sa atong demonstration sa atong nga Acidfast. So at least, aside sa na-explain ko, liwat ya, at least maano nyo gid yan, ma-intake nyo gid, okay, butan nyo na gid sa, hindi nyo gid malipatan. Okay, here, sa atong nga spore stain, aside sa, this is rarely gina-perform sa laboratory, dili maskin sa akon man, sa amon nga school, when namon gina-perform, why? Nga aman. Because when you're talking about mga spore-forming nga mga bacteria, okay, they are highly pathologic and very dangerous. Okay? Kaya kung magkalat na sila, if there is a ano yung term na tama lang, endemic sa, or magwa na siya, okay, ma-infect ang mga tao, okay, with a spore, okay, ayawan ka eliminate sina, aside sa you'll be having a hard time determining what type of bacteria it is. Okay, the amount of antibiotics that you will be taking, okay, para to ensure lang nga mapatay gid siya, high concentration, kay nga man because they are inside the spore, gani ayawan ka mapatyon gid ang bacteria sa sulod mo. So sa aton nga spore stain, okay, sa marita lang, idirektata na lang. Okay, kung hindi mo man sa ma-perform. Okay, your endospore stain is a differential stain using the visualized bacterial endospore and endospore dominant form of your bacterium, which some species of bacteria produces under stressful conditions. Okay, as for mga for nutrition sa gawlat siya nga ma-silo sa iban nga lugar sa diin may nutrients na then dira siyang kabalik sa iyang regular form. Mga high temperature, okay, para hindi siya mapatay dali-dali, and mga dry environments hindi taman ka moist para makamultiply siya. Okay, is forced into a spore using a sting, ideally, with the outer layer of the composed keratin, which resists the stain. The malachite green stain, okay, nabatian yun yung malachite kaina sa aton nga Acidfast, okay, it is a substitute for your methylene blue as a counter stain. Kaysa nga maging blue imong background, okay, you will have a green, okay, ngalan niya pa lang, methylene blue, it will give a blue stain or blue background. Your malachite green will give the stain before sa aton nga spore. Okay, then can be center. It's sa spore tandaan nyo lang. Okay, you have your positions. Okay, kung sa diin makita ang imong spore. You have your central, terminal, and subterminal. Okay, this stain is commonly used to detect spores producing in a genera of your Bacillus and Clostridium. Okay, you have here endospore bacteria and gram-negative or gram-negative, endospore negative bacteria. First, aton nga staining, wala pa. Okay, so they are both clear. Then you have here, steam is used to force the malachite green into all bacteria and endospore. Okay, same procedure nga ginaubra mo sa imong carbofuction, din sa ginainit mo siya hasta maka-form siya sang vapor. Then for your number 3, after mo decolorize, since nakasulod ng imong malachite green sa ano na gid mismo sa hindi lang sa spore but the bacteria itself nga sa sulod sang spore, it will give the permanent green color na. Okay, then ma-decolorize mo siya. Okay, dula ang color green. Then you have your counter stain, which is the safranin. Okay, safranin nga counter stain, which stains all bacteria but cannot stain the endospore. Okay, may color green na siya.
In that,
That.
we will discuss about their
Okay.
properties. Then,
We have
the
here
formation of endospore,
same
its
procedure
structure,
lang sa
and
gihapon.
then about
Okay,
its staining
where
technique.
is area with the smear. Carefully cut towel.
Some
Carefully apply
gram-positive
malachite green to the
bacteria, like
paper
the
towel.
bacillus,
Steam it
which
for
is
10
aerobic
minutes.
bacteria,
And keep the paper
Clostridium,
soaked in the
which
stain
is
for
anaerobic
at the
bacteria,
time. Gently
during
remove the paper
adverse
and with the
environmental
forceps.
condition,
Kagaside pag
during
with
nutrition
paper.
depletion. This
O
bacteria
kasi malakit
will
green,
produce
you'll have
the
it like a green sheet.
that
Okay,
is, the
try ko na
spore
lang kung itaon.
within the
Okay,
bacterial
same
cell.
yapon din.
This endospore is a multi-layered structure which protects the genetic
Kung ano
material
gid man,
inside
next sa
the
tadi.
core.
The spore stain procedure.
The endospores are highly resistant, dormant structures.
Hindi mas namin
They
ito.
dormant
Here you
for
go. Sa
the extended
microbiology.
period, even centuries. They have
The same
no
sa mo nga
metabolism
channel.
and no metabolic activity. So they do not
Okay,
have
let's
role
just
in the reproduction. The process of the endospore formation in the bacteria is referred as the sporulation.
In this video
The
presentation,
endospores
we
are
will see about
highly
the
resistant to
bacterial
the
endo
heat, the
spore.
dehydration,
In that,
the UV
we will discuss
radiations,
about
and
their
the
properties,
chemicals.
then
The
the
resistance
formation of
the
endospore,
is
its
mediated
structure,
by a
and
chemical
then
called
about
the
its
calcium
staining technique.
dipiclinic acid, which is found only in
Some
the spores.
gram-positive bacteria, like the bacillus,
During
which
adverse
is aerobic
environmental
bacteria,
condition,
and the
bacteria
Clostridium,
forms
which
the
is
endospore,
anaerobic
that is,
bacteria,
the spore
during
within the
adverse
bacterial cell.
environmental
The
condition
process
or
by which
during
the
nutrition
endospore
depletion.
forms
This
in the
bacteria
bacteria
will
is
produce
referred as
the endo
the
spore.
sporulation.
That
Later,
is,
the
the spore
mature endospore
within
is
the
released
bacterial
from
cell.
the vegetative cell
This
to
endospore
become
is
the free
a
spore.
multi-layered
When
structure
the favorable
which
condition
protects the genetic
returns, the
material
spore
inside
germinate
the
to
core.
form the vegetative cell
The
back.
endospores,
The
highly
process
resistant,
by which
dormant
the
structures.
endospore
They
revert
remain
back to
dormant
the vegetative
and
cell
for
is
the
called
extended
the
period,
germination.
even centuries.
So the
They have
sporulation
no
helps
metabolism
in the
and
survival of
no
the
metabolic
bacteria
activity,
during
so
adverse
they do not
environmental
have
condition.
role in the reproduction.
The
The
revival
process
of the
of
dormant
the
spore
endospore
into
formation
the
in the
vegetative
bacteria
cell
is
is
referred
called
as
the
the
germination.
sporulation.
It happens by three
The
stages:
endospores
the
are highly
activation, the
resistant to
germination,
the heat,
and
the
the
dehydration,
outgrowth.
the UV
First,
radiations,
the
and
activation
the
stage.
chemicals. The
Here, the
resistance
spore
of the
get
spore is
activated
mediated
by
by
heating
a
at
chemical called
60
the
degrees
calcium
Celsius for an
dipiclinic
hour,
acid,
or
which
while
is found
lowering
only
the
in
pH.
the spores.
No morphological changes happens in this stage.
During adverse
Second,
environmental
the
condition,
germination
the
stage.
bacteria
Once
forms
the
the
activated
endospore,
spore
that
is
is, the
exposed
spore,
to
within the
the
bacterial cell.
substances like
The
the amino
process
acids
by which
and
the
the
endospore
sugars, the
formed in the
germination
bacteria
starts. That
is
is,
referred
the
as the
metabolism
sporulation.
starts.
Later,
Lastly,
the mature
the outgrowth.
endospore is
Here,
released
the
from
swelling takes
the
place
vegetative
due
cell
to
to
the
become the
excessive water
free
intake,
spore.
which is
When
followed
the
by
favorable
the DNA
condition
synthesis.
returns, the spore germinate
Now,
to
we will
form
see
the
about the
vegetative
structure
cell
of
back.
the endospore.
The
The
process
mature
by
endospore
which the
is a
endospore
multi-layered
revert
and
back
a
to the
complex
vegetative
structure.
cell is called
The
the
innermost
germination.
part is called as
So, the sporulation
The core
helps
contains
in the
the
survival
nucleoid,
of the
that is,
bacteria
the bacterial
during adverse
DNA,
environmental
then
condition.
the cytoplasm, the ribosomes,
The
and
revival
then
of
importantly,
the dormant spore
calcium
into
dipicholinic
the
acid
vegetative
and the
cell
enzymes.
is called
This core
the
is
germination.
surrounded by
It
the
happens
outer
by
membrane,
three
or
stages:
the plasma
the
membrane.
activation, the
This,
germination,
in turn,
and
is
the outgrowth.
surrounded by the germ
First,
cell wall,
the
which
activation
is rich
stage.
in
Here, the
peptidoglycan.
spore get activated
Then, the
by
modified
heating
peptidoglycan
at 60
layer,
degrees
called
Celsius
the
for an
cortex,
hour,
which
or
surrounds
by
the germ
lowering the
cell wall.
pH.
The
No
cortex is
morphological
surrounded by
changes
the
happens in
outer
this
membrane.
stage.
This is,
Second,
again,
the germination
surrounded
stage.
by the
Once
spore
the
coat
activated spore
and
is
the outer
exposed
spore coat.
to the
Together,
substances
it
like
is
the
termed
amino
as
acids
the spore
and
coat,
the
which
sugars,
is
the
rich in
germination
the
starts.
keratin-like
That is,
protein.
the metabolism
Once
starts.
this protein coat
Lastly,
is formed
the outgrowth.
over the cortex,
Here, the
spores
swelling takes
gets
place
mature.
due to
Some
the excessive
spores
water
form
intake,
an
which is
additional layer
followed by
called
the
the
DNA
exosporium.
synthesis.
So, this endospore
Now,
is
we
a
will see about
multi-layered
the
structure
structure
which
of
protects
the
the
endospore.
genetic
The
material
mature
inside the
endospore
core.
is a multi-layered and
Among
a
this,
complex
the calcium
structure.
dipicholinic acid
The innermost
inside
part
the core
is called
and
as
the
the core.
keratin-rich spore
The
coat
core
are the
contains
important
the nucleoid,
constituents
that is,
plays
the bacterial
a major role
DNA,
for the
then
resistance
the
of
cytoplasm,
the
the
bacterial spore.
ribosomes, and then importantly, the calcium
So, during
dipicholinic
the
acid
adverse
and the enzymes.
environmental condition
This core
or
is surrounded
nutrition
by
depletion,
the inner
some
membrane,
bacteria
or the
will
plasma
produce
membrane.
the endospore,
This,
that
in
is,
turn,
the spore
is
within
surrounded
the
by the
bacterial
germ
cell.
cell wall, which is rich in
The
the
process of
peptidoglycan.
the endospore formation
Then,
in
the
the
modified
bacteria is
peptidoglycan
referred
layer,
as the
called the
sporulation.
cortex,
The
which
spore
surrounds
formation
the
is
germ cell
divided
wall.
into
The
5
cortex
stages.
is surrounded by the outer membrane. This is,
At normal
again,
environmental
surrounded
condition,
by
with
the inner
nutrient
spore
availability,
coat
the
and
spore-forming
the outer
bacteria
spore coat.
divides by the
Together,
binary
it is
fission.
termed as the spore coat, which is rich in the
But
keratin-like
when
protein.
the environmental
Once
condition
this
get
protein
adverse
coat
or
is
the
formed
nutrient
over the
get
cortex,
depleted,
the spores
the
gets
endospore formation
matured.
gets
Some
initiated.
spores
In
form
stage 1,
an additional
the
layer
DNA of the
called
bacterial
the
cell
exosporium.
condenses
So,
and
this endospore
assembles
is
into
a
an
multi-layered
axial
structure
filament.
which protects the genetic material inside the
Now,
core.
they are called as the mother cell.
Among this, the calcium dipicholinic
In
acid
stage 2,
inside the core
the DNA
and the
divides
keratin-rich
into
spore
2
coat
copies.
are the important
Then,
constituents
the mother cell
that
membrane
plays a major
invaginates
role
to
for the
form
resistance
the
of
4
the bacterial
spores.
spore. So, during
In
the
stage
adverse
3, the
environmental
mother cell
condition
membrane
or
continues
nutrition
to grow
depletion, some
and
bacteria
engulf the
will
developing
produce
spore.
the
Now,
endospore,
the
that
developing
is, the spore
is
within
surrounded
the
by the
bacterial
2
cell.
membrane layers.
The process of the endospore formation in the
In
bacteria
stage 4,
is referred
the
as the sporulation.
peptidoglycan laid
The spore
between
formation
the 2
is
membranes
divided into
of the developing
5
spore
stages.
to form the spore cortex.
At normal environmental condition, with nutrient availability, the spore-forming bacteria
The calcium
divides
dipicholinic
by
acid
the
is
binary
formed
fission.
inside the developing spore, where the
But
calcium
when
enters from
the environmental
out,
condition
and
get
in
adverse
exchange,
or
it
the
loses
nutrient
the water.
get depleted, the
Once the
endospore
protein
formation
coat
gets
is formed
initiated.
over the
In stage
cortex,
1,
the spores
the
gets
DNA of the bacterial
matured.
cell condenses and assembles
Some
into
spores
an axial
form an
filament.
additional layer called the exosporium.
Now, they are called as the mother cell. In stage 2, the
In
DNA
stage 5,
divides into
with
2
the help
copies.
of the
Then,
lytic
the mother
enzyme,
cell
the
membrane
mature spore
invaginates
released from
to
the
form
mother cell.
the 4
Next,
spores.
the special staining. The special staining light,
In
the
stage
Schaefer and
3,
Fulton
the
staining
mother cell
technique
membrane
helps
continues to
determine
grow
or
and
to demonstrate
engulf the
the
developing
endospore
spore.
of
Now,
the
the
bacteria
developing
under
spore
the
is
microscopic
surrounded by
examination.
the 2
By
membrane
this
layers.
staining technique, here, the endospores are stained green,
In
and
stage
the
4, the
vegetative portion of the
peptidoglycan
bacteria
laid
are
between
stained
the
pink.
2 membrane
By this
of the
staining
developing
technique,
spore
we can
to form
identify
the
the
spore
different types
cortex.
of the bacterial endospore. The shape and the position of the spores varies in different species.
The calcium
This is
dipicholinic
useful
acid
for
is formed
classification
inside
and
the developing spore,
identification purposes
where
by
the calcium
identifying their
enters
shape
from
and the
the
position.
out, and
If
in
the
exchange,
position
it loses
and
the
the
water.
shape is the center,
Once the protein coat is formed over the cortex, the spores gets matured.
oval, bulging
Some spores
spore.
form
The
an
example
additional
is
layer
the
called
Clostridium
the exosporium.
viginantans. Next, the subterminal, oval, bulging spore. The example is the
In stage
Clostridium
5,
showii.
with the
Next,
help
the
of the
terminal,
lytic
oval,
enzyme,
bulging
the mature spore released
It gives
from
the
the mother
appearance of
cell.
the tennis racket.
Next,
The
the special
example
staining.
is the
The special
Clostridium
staining, like
tertium.
the Schaefer and
Next,
Fulton
the
staining
terminal,
technique,
spherical,
helps to
bulging
determine
spore.
or to
It gives the
demonstrate
appearance of
the
the
endospore
drumstick.
of the
The
bacteria
example is
under
the
the
Clostridium
microscopic
tetani.
examination.
And
By
lastly,
this
the
staining
central,
technique, here
spherical,
the endospores are stained green, and the vegetative portion of the bacteria are stained pink. By this staining technique, we can identify the different types of the bacterial endospore. The shape and the position of the spores varies in different species. This is useful for classification and identification purposes by identifying their shape and the position. If the position and the shape is the central.
Kaya rin na class, itong nga positioning sa itong nga spore. You have your mga central, your terminal, kag subterminal.
Oval, bulging, spore. The example is the Clostridium
non-bulged
edematans.
spore.
Next,
The example
the
is
subterminal,
the
oval,
bacillus
bulging,
anthracis,
spore.
the causative
The
agent
example
of the
is
anthrax.
the Clostridium
So, by
shallei.
Schaefer and Fulton
Next,
staining
the terminal,
technique, we can
oval,
stain
bulging,
and
spore.
identify
It gives the
different
appearance
types
of
the
the tennis
bacterial
racket.
endospore.
The
Coming
example
to
is
the
the
principle
Clostridium
behind the spore
tertium.
staining technique. It
Terminal,
is done by
spherical,
the Schaefer and
bulging,
Fulton
spore.
staining technique.
It gives the
It is
appearance
also
of the
sometimes
drumstick.
called as
The
modified
example is
seal
the
Nielsen's
Clostridium
technique.
tetani.
That is,
And
it
lastly,
is
the
a
central,
modified version
spherical.
of the Zeal
Ang
Nielsen's
you have here,
Acidfast
mga
staining
bulging
method.
type.
We will discuss the
Lugahabo.
coloring property of
Okay.
the non-spore
The
forming
difference between
bacteria
your
and
oval,
the spore-producing
the
bacteria
shape of
in
your
parallel to each
sa
other.
sulod, yeah.
Once the
Okay.
bacteria
Then
are
there is a
treated with
spherical,
the
okay,
5%
perfect
aqueous
circle, then
malachite
bulging,
green
gahabok.
staining solution,
Okay.
which is
This
the
is your
primary stain
Clostridium
used here,
tetani.
along
Okay,
with
yung nga
the
muni
heat treatment. It gives green color
microorganism
to
na
both the
exhibit sa nga
bacteria,
muning
but
appearance,
only the spore
tetany, that
portion
causes your
of the spore-forming
other thing is
bacteria
tetanus.
fixes the primary
So
stain.
you have your
Next,
muscle
after heating, wash the
paralysis
smear
kung
with
infected
the
ka sa
distilled
imo nga
water. It
tetanus.
decolorize the primary stain easily from the non-spore-forming
Non-bulging
bacteria,
spore.
but the
The
spore
example
portion
is
of
the
the
bacillus
spore-forming
anthracis,
bacteria
the causative
retains
agent
the
of
primary
the
stain.
anthrax.
Lastly,
So,
on
by
use of
Schaefer and
the
Fulton
counter
staining
stain, the
technique,
dilute
we can stain
carbofuchsin
and
that gives
identify
pink color
different
to
types of
the
the bacterial
non-spore-forming
endospore.
bacteria
Coming
and
to
to
the
the
principle
vegetative
behind
portion
the
of
spore
the
staining technique,
spore-producing
it
bacteria.
is done by the
So,
Schaefer and
on
Fulton
spore
staining
staining, the
technique.
endospores
It is
are
also
stained
sometimes
green,
called as the
and the
modified
vegetative
Zeal
portion of the
Nielsen's technique.
spore-forming
That
bacteria
is,
are
it is
stained
a
pink.
modified
With
version of
this,
the
we are
Zeal
coming to the
Nielsen's
end of the
Acidfast
bacterial
staining
endospores.
method. We will
In next
discuss
video
their coloring
presentation, we
property
will see about
of
the
the
practical
non-spore forming
demonstration on
bacteria
the spore
and
staining
the spore
technique
producing
and their
bacteria in
microscopic
parallel
examination.
to each other.
Hope
Once the
the lecture
bacteria
is
are treated with
informative
the
and
5%
useful.
aqueous
Thank you.
malachite green staining solution, which is the primary stain used here, along with the heat treatment, it gives green color to both the bacteria, but only the spore portion of the spore forming bacteria fixes the primary stain. Next, after heating, wash the smear with the distilled water. It decolorize the primary stain easily from the non-spore forming bacteria, but the spore portion of the spore forming bacteria retains the primary stain. Lastly, on use of the counter stain, the dilute carbofuchsin that gives pink color to the non-spore forming bacteria and to the vegetative portion of the spore producing bacteria. So, on spore staining, the endospores are stained green, and the vegetative portion of the spore-forming bacteria are stained pink. With this, we are coming to the end of the bacterial endospores. In next video presentation, we will see about the practical demonstration on the spore staining technique and their microscopic examination. Hope the lecture is informative and useful. Thank you.
Wala gid pakita ang iyang ano. Okay, wala. Kaya procedure gid. Well, that so you have it class. Okay, at least na discuss itong terminal. Dito man sig we have here. This is the live. Okay, yung makita mo sa mga microscope after your staining. Then you have your vegetative na part. Then you have your spore na mga parts. Okay, so wala ka bulge ang ilang mga spores. Then you have your capsule stain. Okay, so you have your picture that you're na ito nga picture kay amo to yapon. We'll have a video presentation, demonstration. With mga plates din sa dalom. You have isa lang ka plate and then you have your catalase nga test. Okay, so go directly. Diretso na lang ako sa ito. Hoping yan. Okay, let me pause na lang. While I'm searching for the video.
Some bacterial species have structures known as capsules present outside their cell walls. Capsules are one type of bacterial glycocalyx. Capsules are structures composed of polysaccharides, proteins, or a combination of polysaccharide and protein. Bacterial capsules can offer the bacterial cells several forms of protection. Capsules can protect cells from desiccation and from host immune defenses. Pathogens that have capsules may prevent the ability of phagocytes to destroy them through a variety of mechanisms and in some cases interfere with the activation of complement and the immune response. Capsules may also play a role in biofilm formation by allowing cells to stick to surfaces and to other bacterial cells. Biofilms are complex communities of microorganisms. Capsules serve as virulence factors for a number of human pathogens, including Streptococcus pneumoniae, Escherichia coli, and Klebsiella pneumoniae. In this lab session, you will be performing a capsule stain. The purpose of the capsule stain is to reveal the presence of bacterial capsules. The chemical composition of capsules causes them to repel most stains, so the capsule will not be visualized directly with a stain. Instead, the staining method relies upon revealing the capsule indirectly. The capsule is also a highly hydrated structure, in some cases consisting of greater than 95% water. Therefore, heating the sample would cause shrinkage and distortion of the capsule. So this staining protocol will be different than those you have already performed, because you will not heat fix the sample. You will follow the protocol for the capsule stain outlined in your NCTC lab manual. Please remember you will not heat fix your sample. The first staining solution used in your protocol contains Congo Red. Congo Red is a pH indicator that is red above pH 5.2 and blue at pH 3 and below. It acts as an acidic dye and is repelled by the bacterial cells, causing the background to be stained. You will place a drop of Congo Red solution on your microscope slide and then transfer your bacterial sample to the drop of Congo Red. You will mix the sample and then use a second microscope slide to spread the dye culture mixture into a thin film that forms a red background on the slide. The slide is air dried, but it is not heat fixed. After the slide is dried, a second staining solution containing Mannavol stain is added to the slide. Mannavol stain is a mixture of acetic acid and acid fuchsin. The acid fuchsin is a basic dye that stains the bacterial cell red. The acetic acid in the stain lowers the pH of the sample on the slide, causing the Congo Red to appear blue. Capsules do not interact with either dye and therefore remain unstained. Upon observation, you will see a grayish-blue background with red cells. If a capsule is present, it will appear as a white halo around the cell. Provided on the screen are the results that you should expect in the laboratory for the 5 species that we have been working with. Do not expect to find a capsule for the Mycobacterium sphygmatis. For all other samples, a capsule should be present. The image on the top right-hand side shows what a capsule would look like in the laboratory setting. The bottom image shows an organism lacking a capsule.
Okay, so there you have it sa ating mga capsule stain. Okay, mas mas summarize or may visual na presentation sa aton nga capsule staining. Pero dito gamit niya ang Congo Red. Pero dili we will be using the grams crystal violet. Okay, so domo ni dan since the Congo-Red is hard to acquire, and atong crystal violet aside sa nagamit sa atong gram staining, okay, this is used for capsule nga staining. Okay, as you can see here, okay, you have here club shell limoni. Then ang iyang background is being stained by your crystal violet, and ang iyang is ang copper sulfate, ang iyang pang gamit niya sa iyang second reagent. Okay, ang staining niya sa iyang crystal violet is for 2 minutes. Okay, do not use crystal violet from your gram stain. Okay, ang iyang doon lang same lang silang crystal violet, but the difference is ang iyang concentration. Okay, you're using 1% of your crystal violet. Okay, so very gently rinse it with your copper sulfate. Okay, ang iyang kung baga ang iyang decolorizer. Because what you will be doing is removing the crystal violet nga ginanos ang imong capsule. Okay, then air dry lang gyapon, then examine mo lang. Now let's proceed sa aton nga TSA. Okay, so TSA is a special type of method or triple sugar ion agar. Okay, or TSI, triple sugar iron. Okay, is another example of a multi-test agar. It tests for the fermentation, okay, with or without gas production, and also for its glucose or lactose or sucrose ang iyang ginagamit. It also tests for the production of your hydrogen sulfide from your amino acid nga production. Okay, you have here the panel red is the pH indicator used in this test medium. Okay, so this is quite complex naman because tanawabla you can test here the fermenting of if ang imong microorganism is a fermenter or not. Okay, if either may gas formation but sa kung wala, kag kung what type of sugar does it use. Okay, now here ang procedure. Okay, obtain a slant of your TSIA. Okay, using the nucleating needle. Okay, stab your assigned organism into the butt of your TSA. Diba? Kung mag-butt kita, you will provide an aerobic nga environment for your microorganism. Okay, then as you drag it, okay, or pagkuha na, then do a zigzag pattern sa imong surface. Therefore, you will be inoculating it sa imong surface. Then, incubate. Okay, the word incubate when you're talking about in microbiology is letting your microorganism grow in your incubator. Okay, incubator is a type of device. You can set what type of temperature you will be having in that certain machine or environment. Okay, diba, Balta? It is a factor para mag-grow ang imong microorganism. Each microorganism have their optimum nga temperature requirement. Then, after the incubation period, record any changes in the tube naman. Because the mga changes sa mga tube will indicate what type of microorganism it is or what does your microorganism does. Okay? Now, you have here review sa mga or mga interpretation kung ano mga changes sa mga TSIA. Okay? So, note the following or remember the following. Okay? First, you have your slant color or sa butt color. Okay? If the slant color okay, indicates there is a fermentation of your lactose and or your sucrose. Okay? Then, if may butt color, okay, ang slant color, dili sa babaw, this is your slant. Then sa dalom, that is your butt. Okay? Kung may butt color, okay, it indicates that it is a fermentation. It ferments glucose. Okay? Then, if may gas formation ba siya or wala. Because if may gas formation sa okay, you will see here na may bubble na this dalom, okay, or may split sa iyang sa imong gidpasudlan sa imong nidol. Then the production of your H2S or your hydrogen sulfide, okay, so is indicated kung may blackening sa imong medium like this one. Okay, you have here sa left, it is an AA, okay, and H2S positive and gas positive. So this is the left side. So you can see here my bubble formation on my crack.
Try new
Yung mga bubble, it's gas
with gentle
positive.
micellar technology.
May blackening.
Today we're
Okay,
going to be doing the catalase test.
therefore there's a
And what
gas.
it is
Then
if
sa
an
imong AA, okay, there is a coloration sa piyak. So kung sa slant gani ang coloration, it ferments lactose or
organism is catalase
sucrose.
positive,
Okay,
it can
then
break
kung
down
wala,
hydrogen
kung sa bata yung
peroxide
coloration like this
into
one,
water and oxygen.
ay it is
So a catalase positive reaction is going
it
to
ferments
cause bubbles,
glucose, therefore it's
whereas a
AA.
catalase
Then
negative
PA naman
reaction is not
for
going to
your
have any bubbles at all.
sa imong dili sa
So
right side.
what I did was I
Okay, your
labeled
catalase is a
the
very simple
organisms out of the first
experiment.
grouping
Naman,
of organisms
catalase
we
test
use
is a
in class,
differentiation
which are
from
positive, and
Streptococcus
those that are negative in
pyogenes
regard
ata.
to
Where
gram-positive cocci.
test that
So we have the
Streptococcus
Staphylococcus and the
pyogenes gid na.
Micrococcus that should be
Ah,
positive,
wait, catalase
and
enzyme
the
for peroxide into
Enterococcus and the
water.
Streptococcus which should be
Wait,
catalase negative.
ito.
Now, if you look at the grand
wala
scheme
bigla.
of things,
Okay, so
most
catalase test,
everything is
diretso na lang sa
catalase
aton nga.
positive except for strep
Pero sa aton nga
and
natural.
Enterococcus.
I-pause na lang ang recording ko na mas makatipid ko tayo.
But in the first grouping,
Kasambuhay ang Nestle Bear Brand ng mga estudyante para sa matibay na bukas. Panoorin ang kanilang kwento.
Ad class. Okay, so
we
this
have
is from the medical lab.
Try new palmolive micellar with gentle micellar technology. Today we're going to be doing the catalase test, and what it is if an
this is from medical lab Lady Gil. Okay, so disclaimer. So she's the one performing it. So it's just for our educational purposes.
organism is catalase positive, it can break down hydrogen peroxide into water and oxygen. So a catalase positive reaction is going to cause bubbles, whereas a catalase negative reaction is not going to have any bubbles at all. So what I did was I labeled the organisms out of the first grouping of organisms we use in class, which are positive, and those that are negative in regard to gram-positive cocci. So we have the Staphylococcus and the Micrococcus that should be positive, and the Enterococcus and the Streptococcus, which should be catalase negative. Now, if you look at the gram scheme of things, most everything is catalase positive except for strep and Enterococcus. But in the first grouping,
Okay, so as you can see here, class. Record to. Shift ko rin. Okay, so record ulit. Okay, so
we have the negative
okay, so as you can see here,
gram negative
ginugaya is wala na siya ng obra sa niyang emulsion. Ay, directly nagkuha siya sa colony. Kaya isa pa rin, this is a disposable nucleating loop. So plastic siya. So usual plastic, hindi mo siya maiihit. Okay? So disposable na siya. Baksa kami tambal. Ano ba na sa colon direkta naman because your catalase test will be using your hydrogen peroxide or Lehman's or commonly known as your agua oxinada. Okay? Your Staphylococcus aureus or Staphylococcus na species may coccus siya. Your Staphylococcus mycococcus siya gamit may coccus. And sa iyang apiyap ni sa iyang left side is a Streptococcus enterobacteriae. Okay? So, this is the differentiation of your staph from your strep. Okay? So, ang normal flora nga sa panit ng gani. Okay, so pwede mo siya matest sa mga panit. Okay, if there is a presence of Staphylococcus aureus, you will be using hydrogen peroxide butang lang sa mga skin. Then, because it is a staph usually sa atong panit, it will cause bubble formation. Kung wala natin principle aton nga pwede ma-perform masin sa inyo nga balay kung may extra ka muda. Okay.
Positive cocci, gram-negative cocci, and gram-negative coccobacilli. So that would be Staphylococcus, Micrococcus, Enterococcus, and Streptococcus are all gram-positive cocci. Then we have Neisseria, which is gram-negative cocci, and Moraxella quadralis, which is the only gram-negative cocci in Moraxella, and Haemophilus, which is gram-negative coccobacilli. So I put an organism on each that should be positive, one that should be positive on here. So I put Staphylococcus aureus on here, and I put Streptococcus agalactiae on this one. So I'm going to add my hydrogen peroxide and look for bubbles. Okay, so as expected, the Streptococcus does not show any bubbles. On the video it looks really white, but there aren't any bubbles. Whereas on the Staphylococcus aureus, look at all those bubbles. Okay, so this is a positive test here for staph, and this is a negative test here for strep.
Okay, so very easy mo na to atong gidyang nga ginaubra sa atong nga catalase test class. This one accept. Sa atong nga 4.1 atong nga microbiology. So there are 3 more topics sa atong nga microbiology. So as we move forward, though repetitive lang na siya. So I'll summarize it as much as possible. Okay, so all videos ang gipakita ko sa inyo, disclaimer, they are though I don't own them. Okay, they are for educational purposes only. So that would be all, class. Thank you and God bless you always.